So next speaker is Dr. Maria Abou Chakra from the University of Toronto, who will talk to us about her work on virtual human development, bridging theory, and experiments.
Thank you. Thank you for having me. I'm assuming you can see my slides.
Yes.
Great. And hi, everyone. And first, I want to say thanks to James, Tom, and Ellen for hosting this lovely meeting. And today I would like to talk to you about an initiative called the Virtual Human Development, which is a grand challenge of trying to create what we call a human simulator. And this is led by Nika Shakiba and Nozumo Yachi from the University of British Columbia and myself. I'm from the University of Toronto. And what we're trying to do is initiate a vision where we can create a simulator that have the ability to predict the outcome of a multicellular developmental system. And this is in hopes that it will not only predict in vitro systems, but also in vivo systems. We think that by just trying to create this embryonic digital twin, we have the potential to change stem cells and maybe even have new regenerative therapies and potentially change our perspective in how we experiment or approach cell development and engineer these cells altogether. So just we have a mission, and we think there are three ingredients. One is an interdisciplinary team, another one is a strategic problem to focus on, so as a group, and the last to standardize semantics and data from all different fields. So our vision is to have this virtual human act as an intellectual sandbox for an international group of researchers, first to understand and build biology together. And this is where the research comes in. And this is going to guide us by a common focus. So whatever we picked our starting condition, whether we want it to be the early blastocyst, we're all going to focus on a particular problem and work on it together. We will collectively develop the knowledge and create a platform that is in line with ethics, legal, and social systems. And this is where the industry partners and policy regulators will come in. And we think that by just trying to build this simulator, we're going to be able to use it not only for clinicians and patients, but maybe eventually also can be used for education purposes. So the past few years, we have focused on trying to build where the research is going to be, what are our milestones, and the types of collaborations. And then the next part is building a community. So I will talk about these efforts today and see where it led us. But before I start, I wanted to sort of discuss why would we need such a virtual system and how can it benefit us? Well, early development is sort of blocked by what we call ethical boundaries. As experimentalists, they can only, grow tissue in a dish, in a lab for only so many set of days. And so we have actually a black box during certain developmental time points. And this is really important because we really do not know enough about it, and we can't access these types of tissues. So what we think is by combining experimental and theoretical efforts, we can push past this ethical boundary, and hopefully we can generate what we call, efficient experiments and rapid research to clinical cycles. Here we've been mentioning them as loops. Basically, you go from research to clinics in a better loop. And we believe that by just trying to combine experiments and theoretical, we can have more informed hypotheses. So our aim is to target congenital diseases such as heart valve or others that can be detected in utero and maybe corrected after birth. And there's a lot of examples where stem cell research is already actively working in this area. So what are the challenges? Well, we know that development is quite complex. It's multiscalar across time. So not only is the system developing in time, but it's also adding complexity. You go from a single cell to two cells. Eventually, you have multiple tissues and organs, and all of them are communicating in different ways. And the way researchers have overcome this complexity is by always choosing their favorite time point or their accessible time points. So everybody's working at different scales and in different time. And we want to figure out a way how we can combine all these efforts. So you can imagine that each time point or each one of these, as the embryo develops, you can pick a different type of cell or different tissue, and each one of them can be our puzzle. And of course, different researchers is cracking away at a puzzle and in hope to finding some ground truth. But what we're hoping to do at the virtual human development is to focus on a single puzzle where we can then apply our various perspectives, whether it's a theoretical perspective or an experimental perspective. And we think by targeting a specific puzzle together, we can be more successful in trying to answer and eventually build this simulator. So this is one of the first steps that we took. We think that by being successful means that we should have a team that is willing to work together. And we actually have been lucky because there's actually about 40 researchers who agreed and want to make this vision a reality. And half of them are experimentalists and the other half is theoreticians. So we're not only combining one type of field, but many fields. We have bioengineers, in vivo experimentalists, we have theoreticians that are working on various types of models. And now we've all agreed to figure out a way where we can integrate in cycles where experimental systems will feed into the model and teach the models, and then the models will feed back into this loop to improve experiments, and it's sort of go back and forth. And I think we've talked about these loops, and right now the virtual human is focusing on these experiment to theory cycles where researchers are talking with other researchers. And we think that's actually the first step to make it a successful digital twin. So why are we doing this? Well, it's actually the perfect time since most of the building blocks that we have or that are in place can be used. We have the experimental tools and data and artificial intelligence already exchanging information and setting up the platforms for learning and making predictions. We have many tried and tested mathematical models that predict complex biological phenomena. And what is key to this initiative is integrating these approaches. We think by working together to tackle the problems using these various tools, we can forge new discoveries and make a working model. And I think what is crucial to the feasibility of this model will be the interdisciplinary approach taken, where theory and experimental expertise are intertwined. And we think that that is the first foundation for a success model. So we want to bridge the right people and we want them to communicate together. So what we set up is we started running discussions that bridges theory and experimental sites, because we think that communicating across fields to understand the various underlying rules and the way we interpret these rules from the various systems would be important. So we've been doing multiple discussions where we find researchers, whether it's a theoretician or an experimentalist, that are working on the same problem, however, approaching it from a different perspective. And then these researchers have the chance to discuss and potentially exchange ideas or interpret their systems in a different way. And I think it has been one of our successful attempts. So as we have been exchanging and understanding each other, we've also been meeting regularly, actually yearly, to identify what are the challenges and what are our next steps. And I know we've heard these a bit before. We do want to establish semantics, or at least identify the models and platforms out there. But I think our group also decided that we don't need to just identify them, we need to interpret. We want to figure out what are different experimental outcomes out there and how do we interpret the different measurements. Can we standardize this? We need to also interpret the outputs and inputs that link experiments and models together. How do we feed this into the platform? We also decided that an importer is important. Nobody's going to recreate a model. A lot of researchers are already invested many years to create their own in their little models. Doesn't matter what language it is. Can we create an importer or interpreter that brings that knowledge into a system? And that way it would be much easier to integrate various work. We also want to create an importer that integrates the various experimental outputs, whether you're generating single cell data or some some spatial genomics. It doesn't matter what can we try to understand how these data work together and how can we use them into the model. So that's actually our next steps. And we've been trying to focus on how to integrate this. And the discussions, I just wanted to kind of give you that we ran, and all the meetings that we've attended together have been quite fruitful. We actually can boast there's actually very few collaborations that just evolved naturally from these meetings. And we think that's actually stating that this project has a potential to go through, because most of these researchers signed up out of their own pocket and out of their own means. And we have the Stem Cell Network, who supported us to meet already 3 times at ISSCR and the Till and McCullough meetings. However, if everybody wasn't willing to sit together and communicate and figure out how to exchange ideas and how to move these problems together, it wouldn't be possible. And it's quite clear that as these projects are growing and more collaborations are added into the list, we will, reach a better successful simulator. We also have initiated a lot of community feedback, and we are hoping to have a voice, especially the voice from a theoretical and a experimental perspective. We have actually made a call for a special issue for virtual human development, where we try to bridge theory and experiment, again, in line with the communication. And we're running a special issue, which actually closes at the end of this month. So feel free to contribute to it. And what we're hoping is that different modelers and different experimentalists can provide their perspective and about the challenges and how they think they would describe moving forward in a virtual human development, and what should the community expect from something like this in the future. So feel free to reach out, because this is a chance for everybody to have a voice. And as you've heard throughout the day, this is an important thing. And last, there's now another chance, another community, which is the integrated multiscale biology. And now we've joined forces, and hopefully we have more individuals who want to become members or help out or integrate in one way or another. So I would like to thank you all for listening, and I hope that you saw a little bit of our approach and our perspective, how we're trying to connect experiment with theory cycles and communicate across various disciplines. I want to thank the Stem Cell Network and Medicine by Design for sponsoring VHD, and of course our advisory board, because they've actually guided us and helped us along the way. And we have a great advisory board that's full of many experts, and we've had different perspective, and without them, we wouldn't have been able to come this far. So thank you all for listening.
Thank you very much for a great presentation. What questions do we have? There are 2 in the chat. Let me read them. Since direct experimentation is restricted beyond 14 days, what strategies or model systems are currently being used to infer or simulate human embryonic development after this stage? Do we stick to stem cell studies?
There are labs that have access to early embryo. I don't know how they get access to them, but these are very limited number of in vivo systems. But most of the time we stick to blastoids, organoids, a lot of the oids in the in vitro system. And I think this is actually the oids, or all the organoids, are going to be our ideal link between models and the real system. I think this is our first step. As theoreticians, I think the oids will create the first step into reaching what we think is happening in humans, because again, human tissue is rare and hard to find. And when we do get it, it's not always necessarily neutral. It might have diseases or something like that. So to try to create these early simulators, we're going to have knowledge from these stem cell models, I think.
I have a quick question for you. Model integration that you mentioned as one of your objectives. We find that extremely difficult, and especially as models become more complex, more detailed. They have a lot of knobs and variables and things like that, and they're not developed with the ability to talk to other models in mind, and that makes it then very difficult. And it also, what makes it difficult is that they don't come with very good documentation and user manuals to quickly understand, what the different levers do, what the different levers are, and what they do in the model. And then I feel like over time, the complexity of these models, when one sees one, it's like, oh gosh, it's just easier for me to build a model from scratch. And so then people just keep rebuilding the same models of the same processes because of the difficulty to use the existing ones. How do you approach this?
Yeah, so we will use existing ones from the researcher who's going to first provide it, right? So this is where, I mean, as a modeler, I don't want to recreate mine in Python if I've already written it in a different language, right? So that's kind of my problem. And I think everybody else has the same feeling. I've already spent 10 years creating this model. I'm going to continue it. But lucky for us, we have James and others who've built platforms that may make it easier to import. And I think that's kind of the idea is, let's say I did write mine in whatever language I have, maybe now I can import. And I use Mathematica, don't judge. But I think it's a great platform. But I can import Python, I can import Julia, I can import R. And I have a feeling that that's probably going to be the future of these digital twins. I can't imagine that each one of us is going to recreate our model, but we're going to prove that it's valid, prove that it has a base in whatever question that we're trying to answer. And I think researchers will appeal to something like this a lot more than if I had to make you rewrite your code in some other language, to be honest. If I've already worked on mine for more than 10 years and others have done like 20 years of work on their systems, it's going to be hard to convince them that they need to rewrite it in a certain platform. But I
Well,
think
it's
yeah,
yeah, it's more about, so the languages, that's one challenge, but I think that challenge has been pretty well addressed nowadays with SBML and other standards that can exchange some of these models, at least molecular level agent-based models. That's a
right.
different story. But it's more about the conceptual structure of the model. What's in the model? How do you use it? It's like, imagine getting into a spaceship without being trained on how to operate it. That's how some of these models can feel.
True. And I agree with you. I don't think I could use a model or somebody else's model without, but I think we are working, this is why I focus on researchers and community. I think the first step is that we are working with theoreticians who are all coming from different point of views of models. And I think if as a group, we start to learn from one another and we decide on what is the first building blocks of this model, what are the things that we need in this spaceship in order to have it running before the external community comes in, right? I think this step has to be done between the group, if that makes sense. I mean,
Yeah.
James is actually one of our advisors and members, and he, to me, he's a pioneer in all of this. He's created platforms and so we're going to leverage on existing systems and programs and platforms. But I think the first step is we have to talk to each other. Even modelers have to talk to each other, and this is why we spent a lot of years, because we all interpret what we even mean by model differently, and what do we mean by which scale. And I think we've all agreed that it has to be modulable and it will have the right semantics to be integrated properly. And if later we decide one steering wheel is better than another steering wheel, I think it will be, sort of like a functional platform where we can hypothesis test. So it's not going to be like set. So if later we decide that we don't want steering wheels and something else came into play, it should be modular enough that we can test these and then move forward. Or you can include or not include certain aspects of development. I can't imagine right now we'd be able to run a whole embryo, even given the best infrastructure out there. So we're going to have to make it modular. We have to be able to target certain time window, certain organ. All of these have to be integrated somehow. And we also probably should create a bypass system. Like if you're just going to look at the lung, maybe you do not need everything else developing out there. And we would have some sort of a mean simulator that gives you a starting condition. So those are the stuff that we are discussing right now. But you're right. It's not going to be easy to just use somebody else's model, but I'm hoping if researchers agree together, then maybe we could come out with something that is valuable.
Thank you.
I wanted to jump in. Maria, could you go back to the slide where you had your 4 aims for integration and development? Because I found those very,
yeah.
Yes, this one. Are you thinking here actually of trying to do automated model generation from experimental data? Is that what number 2 is about? Of the little bit of the kind of thing that Sarah Teichmann was talking about just now? Or maybe you could just walk us through a little bit more of how you interpret these.
So there's more than one level, right?
Yeah.
So right now, I think we have a few people in our group who can use AI to now decide a lot of to predict some of these models for them from the organoid data. And I think we're going to start using them because we spend a lot of time guessing or learning or having this knowledge base. But that's one aspect where we're going to figure out how to interpret the outputs so they can go into the model. But also, we want to be able to, if I want to plug in at a certain time point in our system, I want to be able to use maybe single cell data or some other, because now you have GFP data and there's so much data out there, whether it's image data or anything else. But how do we interpret these data so they can even work together? And so right now, I think what we want to do is figure out the outputs of all these experimental systems and how do they become inputs. So if I want to start at day 20 instead of day 1, or I want to start at a different day in embryonic development, how do I interpret the data that is coming out of experiments? And then it becomes my initial condition for my models. There's two levels, right? So there are groups, and I could say like Linus and Guillaume in our virtual human. So Linus and Guillaume are working together. He has an organoid system, and they use AI to try to pull out what they think is the interpretive model of it, right? So and try to see if you can repredict. And I think that is a great version of this interpreting the output so they can go into the model. But we also have so much other data and databases out there, and we're hoping to try to leverage them and somehow make sense of them. And I'm guessing it's going to have to be some form of AI model that will try to interpret it, some sort of training that can be then put into these models.
I mean, because one thing that always comes up, and I think the previous talk was beginning to address this, is that classic omics is endpoint measurement. So you don't get true time series. And you don't necessarily know what the pathways are. And so when we do dynamic models, we typically start with the idea that we know the gene regulatory networks, we know the signaling networks, although Gary talked about trying to generate perturbations of those with AI. And given those networks, then we try to understand control. And we face 2 big problems. One, that the omics doesn't tell us what the networks are.
Right.
And it doesn't tell us what parameter values would be for those that were networks realized as dynamical systems. And then we also don't really understand how to connect the molecular level with the cell behaviors and higher level phenotypes that we care about. And so I think if we can use machine learning AI to try to make those correlative connections, that would be fantastic. And I was wondering if that's something that you've been thinking about, and whether you think it's feasible, what you think the key challenges are in that direction.
Of course, I think it's feasible. I think that's where we're heading. But there's going to be a lot of challenges. I mean, first, we won't have any training, a lot of training systems. So it'll be a different approach than having all this data and then predicting. I think this is going to be, well, I don't know about too much about the AI but basically you have a data set, usually train on it, and then you predict using a different data set. But here, if we're trying to learn some sort of a model, we might want to use as much of the data as we can. And so then our predictions are not going to come out of the AI. It might be like the way the model interprets what we learn from it. And so there's just going to be a different loop, as just to keep going with the loop. So we will have like an AI model versus, experiment loop, and maybe then feed it back brand new data. Yeah, I think the challenge is one thing that I think about AI is that it either overfits or underfits, and you can only predict within the data you give it to. And the beauty of certain mechanistic models is that you're not really usually limited to a certain time. And I want to make sure that we still maintain that possible predictability that you can push outside of the box of data, right? Because that's kind of the big problem with these early systems. We only have data to whether it's day 14 or day 15, and then there's a black box. There's nothing until birth. and then you do not get data from birth, right? You only get it if something is wrong. And so we need to fill in these gaps. And again, you can correct me if I'm wrong. I believe that AI's ability to predict outside of its, interpretable zones might be hard. And unless we create the right mechanistic models and right interpretations and hypotheses and other types of systems, I don't think we're going to be able to push past that boundary of wiggle room. I think others can chime in. I don't use AI as data. I'm really purely theoretical modeler, but I think it has a lot of use. Like for me, it would be more reinforcement learning. I think that would be our next. I think models can benefit from reinforcement learning. Like we learn different strategies, what cells can do. I think we can push those, but that could be because I come from a game theory background, so there's a lot of overlap there, and more like an evolutionary model. But we will have to integrate a lot of different things to push from knowledge, AI data, or our experimental data, and things we don't know. And that's where this development is. There's a big black box, and not even textbooks can fill them.
Okay, thank you.
Thank you. Thank you for giving me a chance to introduce this initiative.
Thank you.